Hello and welcome to another session of online learning. Okay, so I know I didn't assign these top problems on your homework, so let's start here with the homework. I know I didn't assign these, but I want to kind of go over them with you, just to kind of just review what we talked about yesterday. So how are traits controlled through multiple alleles different from those controlled by complete dominance? So remember, with complete dominance, we could have capital R. We could have something like capital R, lowercase r, or we could have lowercase r, lowercase r. And so basically we have three different genotypes. How many phenotypes is that? Well, this would have been red, this would have been red, and this would have been yellow. So two phenotypes. So you'd have red or yellow. Those are only phenotypes. And with multiple alleles, basically we can have an infinite number of alleles. Remember I said HLAs have 100 different alleles, so that's a lot of different combinations. I can't even go through all of that in my head and figure it out. It's crazy. And so then codominance, so incomplete and codominance both are kind of the same. I'm going to address both of them, even though the question says just codominance. But RR would be red, just like we talked about. RY would be, now in codominance, technically that would be spotted. Like it would be red with yellow spots, or red with yellow stripes, something like that. Incomplete dominance, it would be orange. And then YY would be yellow. So here we have three different phenotypes and three different genotypes. But again, that's it. That's the maximum, three. And we know that with human traits, we have way more than that. Like with blood typing, we look down here at this next question, and we do have the different phenotypes for blood type are A, B, and O. And if we look over here, the genotypes would be IA. So that's just basically is AA. I think you guys were taught by Mr. Bronson, and this is totally fine. Actually, this is the way I do the problems. You just write AA for A blood type, and then if you're going to do heterozygous A, it should be A. Oh, remember, heterozygous A is AO. O is recessive. It's not AB because AB is a blood type. They're codominant together. So for B blood type, that's IB, or IB lowercase I, and again, that's because B is dominant over O. So probably the way you're more familiar with writing this is like this. And why do we even have to bother with these letters over here, the IAIA? Well, do you know which way it's going to show up on a test? Like, for example, if you're taking the ACT or SAT, if you're taking the end of course exam, do you know what it's going to look like? No, it could be either one of these methods. As a matter of fact, I think these ones here in the middle right here are what we've seen more commonly. So AB just as IAIB. I mean, you could technically write BA, but why would you confuse yourself that way? Just write AB. So there's AB. And then type O is lowercase a, lowercase I. So you could also write it lowercase o, same different. Remember, just this O stands for immunoglobulin. Okay. And things went cattywampus down here. Let me see if I can get this back to close enough. Okay, so a male is heterozygous A, so we should have an A, and over here we should have an O, and on the side here, which it's not going to let me write over here very easily, that is just wonderful. When things don't work out the way you like them to, just do your own thing. I don't like the way those boxes are working. So, there we go. And I am just going to copy this, and that way I can just paste it every single time I want to do this. Copy. All right, so what am I going to have here? So heterozygous A, and then O. And again, it could be IA with a lowercase I, but so heterozygous A is A and O. Why is it not AB? Well, because AB is its own blood type, so it's A and lowercase o. And then the female is homozygous B, so homo means same, so we got two B's right there. And so what should we have here? All right, and see again how we get those connected there. So AB, how do we write our results? So 50%, and again, you just have to give me the results of what's there. 50% are AB, and 50% are B. It's B blood type, not BO. We're just talking about the percentages here. You don't have to worry about any types of ratios or anything like that, okay? And I'm going to keep messing with this until I get the format right, probably, so. Insert my own table. Yeah, see? Yay. Okay, so male is type O, so we're going to have two lowercase o's. Female is going to be AB, so we're going to have an A and a B. And so here we have AO and AO and BO and BO. All right, so what do we end up with for results? Again, two of the boxes are A, so 50% A and 50% B. The genotype is AO and the genotype is BO. All right, 6. All right. Let me just start deleting some of these boxes real quick. All right, so 6, we should have A and a B. Remember, only one letter per box. And so here we're going to have an A and an A, so that's type A. We're going to have an A and a B here. Here we're going to have an A and a B. Again, if you wrote BA, fix that because you're just going to confuse yourself. All right, and so there you go. What are the results for this one? All right, so we have one box with AA, so that's 25% A. We have two boxes with AB, so that's 50% AB. And we have one box with B, that is 25% B. All right. Hey, there's number 7. Okay. I don't even know where the chart went. I don't care. There we go. All right. So heterozygous B. What do we put for heterozygous B? Hetero means one of each, right? So we got a B and an O. And type O. Remember, type O is recessive, so you have two O blood types. So we put a B and an O there. A B and an O there. And over here we're going to have OO. And over here we're going to have OO. Oh, not 0, 0. OO. All right. So what is this? And this is how many boxes have B? 2 of them. So that's 50% B. And again, we don't have to put down A, B, and A. I mean, technically, probably you should because we know that there's 4 genotypes. But for these ones, just to make things simple, we're going to just tell me what's there. And 50% of the offspring will have type O blood. Okay? All right. Here's problem number 8. Okay. So, conduct a cross between a man who is heterozygous for type A blood and a woman with heterozygous B blood. So, hetero means one of each, right? So, AO. And she is heterozygous for type B blood, so that's going to be B and O. Again, one of each, and it has to be one dominant, one recessive. It can't be AB because AB is AB blood type. They are A blood type and B blood type. So what do we put here? A, B. All right. Oh, I put a 0 up there, not an O. There we go. All right, here we have B and O. That's B blood type. Here we have an A and an O. That is A blood type. And here we have OO. And that is O blood type. So we're going to have 25% of each. 25% AB, 25% B, 25%, let's see, A, and now we have 25% O blood type. All right, last one. Delete table. There we go. All right. So, conduct a cross between a man who is AB blood type. Pay very close attention to this problem. If you've been tuning out, pay close attention because this one has a common error that a lot of students make. So here we have AB and a woman with heterozygous A blood type. So AB is AB. Remember, heterozygous is an A with an O. So here we have AA, here we have AB, here we have AO, and here we have BO. All right. Now here's where students make the mistake. What are your results? How many individuals, or what percentage will have type A blood? And actually right there, that's a mistake. Answer the question. So I have a lot of students tell me that 25% have type A blood or 25% have AA. All right, so this one right here is type A, and this one right here is type A. There's no different, they're different genotypes, but the phenotype for type A, they're both type A, so it's 50% are type A blood. The genotype doesn't, like, you can't differentiate between the two. It's kind of like if you're dealing like with a brown rabbit and white rabbits, and let's say that white rabbits are recessive. Well, if a rabbit is heterozygous for brown and a white allele, and another one is homozygous for brown allele, you can't tell the difference between the two. Like, they both are really they're both the same in our eyes. Like, we see them both as brown. All right, you can't tell the difference, and so like you can't tell the difference between this person with A blood and this person with A blood. Like, their blood cells are going to have the same protein on the surface. All right, so if we have so right here, what's that? What blood type is that? Well, that's AB, and there's only one box with AB, so 25% AB. And then the last one here, this box right there, that's BO, but that's the only box that has anything with B in it. So it's 25% B blood type. Okay. All right. A lot of times you'll see problems with this where it's asking a question like, so and so had children at the hospital, and there was a mix-up, and then you can prove which baby belongs to which parent without doing a full genetic test. You can just run a blood test, and that's usually a really easy way to figure it out. For example, if both parents have AB blood type, there is no way possible, even if one parent has AB blood type, there's no way possible they're going to have a child with type O. I mean, just look here. We just did a cross with one parent with AB blood type. And notice there's no way possible he could have a child with type O blood, because the only way you can have type O blood is to have both parents at least be a carrier for the trait. Or like here, this parent is a carrier for the trait in number 7 for type O blood, but the mother had type O blood as well. So, all right, we're not going to worry too much about the rest of this down here. It does talk about agglutination, which we're going to review in today's lesson. So, alrighty. Let's move on. And so again, the beginning of these notes are pretty much so just review. Here it's just how do we do these Punnett squares. I just went over how to do this on your homework review, so I'm not going to bother with reminding you of that. Remember, if you have type A blood, it has the A antigen. An antigen is an antibody generating protein. It's a protein that identifies it. So, for example, if you're type A blood, your white blood cells don't recognize type A blood because it's you. It doesn't want to attack you. All right. Now, you might say, well, when in nature are we going to come across a different blood type? Well, you're not really. But the point is, another person's blood type would come across, and it's let's say they have type B blood, and they're going to have a B antigen, which your body just doesn't recognize it. And so your body's going to basically, your immune system's going to flip out and attack those blood cells. Now, what it's meant to do is to attack, like, for example, bacteria and viruses have proteins on their surface, and those proteins alert our immune system, and our immune system flips out and kills those, which makes sense. But your body is seeing this helpful substance, blood, as being harmful because it has a protein on the surface that doesn't match your proteins. This is a major concern, for example, with organ transplants and such as well. Matter of fact, we've got to be even more specific. There are more antigens to take into consideration, like, for example, if you're doing like a liver or heart transplant. So the A blood type has the A antigen on the surface, and so it produces B antibodies to attack it. There's no such thing as an O antibody because O doesn't have a protein on the surface. So A has the A antigen, it produces the B antibodies. B has the B antigen but produces A antibodies. So if a person with type A blood is taking blood from a person with type B, while they're producing the anti-B, they're going to attack B. Or, for example, if this person has AB blood and they're donating blood to the type A blood person, again, they have anti-B, they're going to attack anything with B in it. That's an easy way to think about this, is type A has the A antigen, therefore it has the B antibodies. It's going to attack anything that has the letter B in it. It can receive blood from a like blood type, type A, or from type O. B can receive blood from B or O. Why? Because it produces the anti-A and will attack anything with an A in it. So both A and AB are out for a person with B blood type. AB doesn't produce either of those antibodies because, again, it would be attacking itself. So notice it can receive A, B, AB, and O. It is a universal recipient. Now, type O has none of the antigens. It has no surface proteins. So therefore, it's just kind of like this neutral blood cell that has no surface proteins to identify it. So it produces both of those antibodies. So yeah, if you have type O blood, you've got to get type O blood. That is your only option. You cannot receive blood from any other of these blood types. Now, that's kind of why type O blood is so useful, because look at type A receives O, type B receives O, type AB receives O, because there's no antibodies to attack on type O. So a type O is a universal donor. It can donate to anybody. But if you're type O, you can only receive from another type O. So that's why type O blood is very useful. Now, what's going on with this whole positive and negative thing, right? A lot of students are asking that, what happens with if I have like A positive or A negative? And this is really getting down to where human genetics really starts to expand. And your understanding of it starts to expand. And this is what we call a polygenic trait, meaning there's more than one trait that there's more than one gene producing these antigens. There's another one. So what they found was that when you did blood transfusions, you could give A to an A blood type person, and it usually was fine. But like, for example, Al could give blood to Andrew, worked out fine. But if Andrew gave blood to Al, things didn't go so well. And that's because of a second protein called the Rh protein. We actually first discovered it on the rhesus monkey, which is where the term Rh comes from. So if a person has the Rh protein, then they have the secondary protein that's there. And so that can cause some problems. So if you're Rh positive, then you have, sometimes they call it the D antigen. Rh antigen, D antigen. Sorry that it's not just one thing. I've actually seen it even a few more other names. Yes, I wish it was more universal, but it doesn't work that way. So if a person's Rh positive, they have the D antigen. So in my previous analogy, Al could give blood to Andy, but Andy couldn't give it to Al. So what happens here is that Andy was Rh positive. He has the D antigen, also known as the Rh antigen. So he doesn't produce either of those antibodies, and he can receive both positive and negative. As long as the A, B, and O blood typing matches up, he's all good. He can receive it from anyone because the people who are Rh positive don't produce either of the antibodies. But if you're Rh negative, like in the case of Al, there's no D antigen. And so although the A works out great for him, he produces this anti-D antibody. And so if he receives blood from Andy, Al, down here, produces this antibody, which will then attack this positive protein that's on the surface. Now, actually, what they found is that it's a little different with the sensitivities on this. It's more of an immediate response with the A, B, and O blood typing. The RH one takes a little bit more of a timed response, and we'll get into that in just a little bit here. So here we go. Here's blood typing, and when I'm standing up in front of class teaching, I like to cover some of the stuff up, and I am not doing that right now. So try not to look at the answers over here. And now that I've pointed them out, I'm sure you're going to look over there. So look at right here. So what they did was they figured out a way to do blood typing. You could take the antibodies and harvest them and put them in like a little vial. And what you can do is you can drop some blood on a plate, on a smear plate. And so you drop the blood down, and then you put the antibody A on it. And if it clumps up, that tells you, since antibody A attacks the A antigens, it tells you that this sample right here has the A protein. This side over here has the anti-B protein, or the anti-B antibody. And so since it clumps here, we know that it has the B protein. If you see clumping, the protein is present, because this right here was given the B antibodies. This was given the A antibodies. So over here, this particular sample would be AB. How do we know it's AB? Because the A protein is there and the B protein is there. This one, nothing happened here. So is the A protein present? No. Is the B protein present? Well, we see some clumping, so we know that tells us that agglutination occurred, so therefore this person has B blood type. And this is how they test your blood for blood types in a lab. All right, so this one clumped with A, but not with B, so this blood type is A. Neither clumped. That means no proteins. Think of the O as like a 0. This is type O blood. There are 0 antigens. Nothing there. No antigens. Something like that. And so since we see no reactions, that tells us it's type O. So when we do this in class, I will give students a little plate, and we'll use simulated blood. We don't want to use real human blood in a school setting. But what I have students do is I have them put a little dot of blood in the upper right-hand corner of the plate so they can see what a non-reaction looks like. And so that's what this is right here. This is what the blood sample looks like if it does not react. So if we treat this one with antibody A, it clumps. That means A is present. So it's A something. All right, here we have the antibody B, no clumping. So it's just A. There's no B there, so it's A. Now, anti-D, again, this refers to the Rh antigen. You can also refer to it as anti-Rh. And so this person has clumping, meaning they have the Rh antigen, which means this person has a positive blood type. All right, so here we go. We got this one. This is the sample right here. Right here, no clumping under A, so that there's no A in the results. Clumping in B, so there's a B. This is B positive. This one clumps in all three. So it has an A, it has a B, so it's AB, and it has clumping in the RH, so that means it's AB positive. Down here, it is nothing under A, nothing under B, nothing under the anti-D or RH. So this is O negative. Negative means there's not an RH protein. So a couple things about this. Transfusion reaction, we talked about that. You can't give the wrong blood type to a person. So let's just think about this. A person has A positive blood type. What are some blood types they could take? Well, obviously, if they're A positive, they can take A positive, right? Could they take A negative? Yes, they could take A negatives. Because they have the positive protein, that means they aren't going to produce the antibodies. They can take the negative blood type. I have a game that with a web link. I'll kind of work through it with you a little bit, but I want you to go through that game and play it until you basically have it down and understand what's going on here. So it's going to be your way of testing blood types and understanding this correctly. Hemolytic disease of I had a moment of ADD there. Before we move on, so A positive, what blood types can it take? A positive can take A positive. A positive can take A negative. So are there any others? Can it take AB positive? Well, no, because they would react with the B in it. And that means that obviously AB negative won't work. B won't work, that's for certain. Neither of the B's will work. O, could it take O positive? Yes, it could. Could it take O negative? Yes, it could as well. So, hemolytic disease of the newborn is also known as erythroblastosis fetalis. And basically what happens is the mother's antibodies attacks the baby. And this happens if mom is pregnant and she is a negative blood type. For example, my mom was O negative. I am A positive, and as I was born, at some point in time, there was mixing of my blood and her blood. I mean, if you've ever seen a birth, blood happens. Okay, so anyways, and so you're supposed to be given a shot, and it acts as an immune suppressant if the baby has an incompatible blood type. And so it keeps you from producing the antibodies to attack in case you become pregnant again. And that's actually exactly what happened with me and my brother. My brother is also A positive. My mom is O negative. Their blood types were incompatible. So because she was exposed to my blood during childbirth, she built up antibodies for the blood cells, and the antibodies can pass through the placenta and into the amniotic fluid, and actually were attacking my brother, and they nearly lost him. He ended up he was born 2 months premature at the very edge of what was acceptable to be born at the time. He spent first couple months of his life in NICU, which is a neonatal intensive care unit. And so, yeah, it's something that happens. Now, you shouldn't have to worry about this. Even when my mom was having children, it shouldn't have been a problem. I'm not exactly clear as to why she didn't receive the shot or what went on, but this is something that can happen. It is a medical thing, and so this is the justification for why you do that. Any of you who are interested in going into the medical field, this is something you're going to run across, and this is you need to make sure that this shot occurs, so that way you don't have any complications with further children. That's why there is only me and my brother and my family, aside from my parents, because they had to stop because the second pregnancy was so high risk and so difficult. All right, so again, here is the web link. The web link will be on the Google Classroom page, but I want you to go to this website and practice doing this. So let's go ahead and take a look at this real quick, and then we're done. So it should be queued up. You probably are going to have to click on some of the options up here. This is in Firefox, not in Chrome. Sorry, I use Firefox for reasons. Just don't worry about it, okay? But anyways, so here is the link. Why don't you click on the link? Here's the page, and we're going to proceed. So we're going to do a quick game with random patients. You can do the other ones if you want to. Sorry, the noise is kind of high-pitched and annoying. So we have to select our patients. We got this girl here with the purple hair and really strange, thick double eyebrows or something going on there. We got this dude here with the Fu Manchu. And we got this lady, I don't know, she looks elderly-ish. So go ahead and pick one. I'm going to pick the dude with the Fu Manchu, just because it's kind of cool. Now, you're supposed to poke the person in the arm. If you poke them somewhere else, it's kind of fun to do. All right, I'm sorry. Anyways, so here, let's draw their blood. All right, now, we're going to put some of the blood in the A vial. This is antibody A. So if we see clumping, we see clumping. So what blood type is that? I'll put it in the B. No clumping. Okay, so that, all right. So what blood type do we have so far? So you should say, and you can click on it for help, that this is A blood type. It's A something, because B doesn't have the protein, so we know that the A antigen is there. All right, let's see RH. Nothing. All right, now all of you guys are going to have different people, and you're going to have different results. So do this until you can get a perfect score. So let's go A. All right, because I know that it's A. And is it positive or negative? Well, there's no clumping into RH, so that's negative. And nothing like a good British swear. All right, so you're bloody right. All right, so let's move on. Blood transfusion time. Okay, what blood types can we give him? What did we say he was? Oh, right here. A negative. All right, so which blood types can we give him? And I think since he's A negative, we can give him A negative. Oh, that makes him happy. Oh, but he needs another pint. So, maybe you're saying, well, I think he can get A positive. All right, well, let's just see. Can A positive work? Should it work? I think it will not work. Oh, ooh, don't do that. That's not good. Don't, no. All right, remember, O is a universal donor, so O is always good. Question is, O positive or O negative? O positive? Ah, no! All right, well, O negative is the only other option for this guy. Should have been yes! A negative and O negative should have been the two that we should have done. Now, I'm obviously not going to score very well on this because I'm demonstrating for you how to do things wrong. I want you to go through this, do it correctly, get 5 stars, and once you get 5 stars at the end, if you can, take a screenshot and post it, and that's your homework assignment, is to post a screenshot of you getting 5 stars. All right, and that should be everything.